The science of biology. Louis Pasteur said, science is a way of thinking, much more than a body of knowledge. So if you have your spiral notebook, go ahead and open up your spiral and so you can take notes along the way. So what is biology? It's a scientific study of life. All living things have a set of properties. They have order, which is a hierarchy that begin as cells, then they can move up to tissues and organs. They respond to stimuli. That means environmental conditions. Reproduction, they're able to pass genes along. Adaptation, they're able to be successful in their environment. All living things also share these sets of properties. Homeostasis, that means they have a stable internal environment that is needed. It could be a temperature or pH, or they have to have a certain electrolyte level. They have a way of processing energy. It could be from the sunlight or something that they have consumed. Growth and development. Evolution, they're going to change over time. What characteristics of life are demonstrated here? So we have a Venus flytrap. It is open. You can see that it's sticky inside and it does have prickly things that will attract that fly and then close on it so it can eat it. What characteristics of life are demonstrated here? So the polar bear has caught a seal, killed a seal, and that seal will then be food for the polar bears. So, number one, organization of life. The biosphere includes all parts of the Earth. That's the surface, the atmosphere, the ocean, and what it has in its different layers. So in the ocean, of course, we have fish and algae. And the atmosphere, the lower area, the troposphere near us, we have birds flying around, for example. An ecosystem includes the biological community and the surrounding physical environment. So we're looking at this image here. We have a lot of different animals that are visiting a watering hole. And so this would be an ecosystem. We have different types of birds. We have zebras. We have elephants. We probably have fish in the water. The biological community is made up of all the population living and interacting in that area. So everything that's interacting, even if some of those items consume some of the same items in that ecosystem, they're all the same. They're all part of that community. A population includes all the members of a species that live in the same area. So we're going to see the shaded area here. We have all of the elephants. So that's a population of elephants. An organism is an individual life form. So before, you could see the shaded area was the elephant in the water and the elephants walking in the background. That's a population of elephants. But if we're talking about just an organism, that's the individual elephant that's walking in the water. So an organism, of course, is an individual life form. And a lot of this is review from past life science courses. We have our organ systems. We have a digestive system, a reproductive system. We have different organs. It could be a liver or a heart. We have groups of tissues. It could be skin tissues or muscle tissues. And then we have the cell, the smallest unit. What levels of organization are shown in these pictures? So we have different types of liverworts on the side, and we have a microscopic image of a leaf. So looking back at our levels of organization, we're looking at different tissues, plant tissues. Number two, scientific method. We have a process in biology where we have to gather information and follow certain steps. So the scientific method, of course, begins with asking a question. It has to be something that we can test. It may be as simple as, what type of bubble gum makes the largest bubble? Could be something as simple as that. That's your question. And so you're going to do a little research. That might mean that you need to chew a lot of different types of bubble gum up, right? You're going to construct your own hypothesis. You may say, I think that Hubba Bubba Bubblegum is going to have the largest bubble. And so you're going to compare Hubba Bubba, maybe Wrigley's gum. You're going to test it out. And so this is how many of our famous discoveries were followed was through the scientific method. One example is the smallpox. So smallpox, we don't hear about it a lot anymore because it has been eradicated, but it was a deadly contagious disease that had a 30% fatality rate. So we have Dr. Jenner, and Dr. Jenner was observing that dairy maids, which were people that are working and on a farm and they are getting milk from the cows, they seem to be protected from smallpox. And so he had this observation, and so they had been around cows that had cowpox, very similar to smallpox. And so he's trying to see, is there some kind of correlation there? So we have a hypothesis, which is a testable prediction. So his prediction is if a person is exposed to cowpox, they would also be immune to smallpox. That's his hypothesis. So he's going to experiment a little bit. So we have different types of experiment variables that can jump in. We have an independent variable, which was a new factor that can be introduced and tested, a dependent variable, which is a measured result by the independent variable, control variables, which are kept constants, they don't influence the dependent variable. So Dr. Jenner is going to inoculate, which means give the shot to an 8-year-old child with cowpox. Then later, smallpox. So our independent variable is the vaccine. The dependent variable is the immune response to smallpox. The constant is unexposed children. So they're going to use some children that haven't been exposed to anything. That's going to be the constant. So many experiments were done for the comparison. The control group does not receive the independent variable. The experimental group, though, does receive the independent variable. As we see in this image, we have two twins, and one has smallpox and the other does not. So you're going to have data. And so the data was the boy was inoculated 20 times with smallpox without catching it. You're also going to have a conclusion. So he's going to conclude his hypothesis and how it was supported. Then you're going to have a peer review. So this involves publishing your results to other scientists in that field so they can review it, check for errors, see if there's any bias, and any uncontrolled variables. You may have a margin of error, which is an estimate of how different a result is to the actual variable. Here's Dr. Jenner. He's able to locate several other parents who were willing to volunteer their children. The results were finally published, and he called his technique of vaccination VACA. So you're going to have a little bit of experimental design. It could be a natural environment where you're taking place in the real world without manipulation. It could be similar to how monkeys are observed in the jungle. You could have a controlled experimental design where labs are going to be done in an artificial environment, in a room, in an office. Bias is a preference for an experiment to turn out a certain way. So we don't want to have bias while we're doing an experiment. We don't want to be paid to do the experiment and we want it to have a certain outcome and things like that. Or maybe you don't want the outcome to be a certain way because it would protect your job if it didn't work out that way. You could have a blind experiment where you're trying to get away with the placebo effect. So you have 20 people and you're going to give 10 people a certain pill to see what happens. You're still going to give the other 10 the same pill, but it might be a sugar pill. Double-blind experiments really prevent both scientists and subjects from even knowing who was given what. So we're going to watch this experiment here, The Secrets of Psychics. and identify the following. We're looking for the dependence, the independence, the control, the experimental groups, what the sample size was, and the probability of success. If you want to, on your paper, go ahead and make 4 boxes. So one box is going to be dependent and independent. I think in that box, I would even separate it into columns with a D column and an I column. And then our second box is control and experimental, and you may want to do the same. Have a C column and an E column. What is the sample size, and what is the probability of success?
My investigations have taken me to dozens of countries, but until recently, one of the most interesting wasn't open to scrutiny. It's long been rumored that Russia harbors psychic talent like nowhere else in the world. The country has an ancient mystical tradition.
Good morning.
Good morning,
And the government has encouraged psychic research.
To Moscow?
That's right.
Any luggage to check in, sir?
2 pieces.
Now that the country is open to visitors, I wanted to check things out for myself. I'd seen the films that came out during the Cold War. Juna was a famous healer who had Brezhnev as a client. Karl Nikolaev could apparently turn on lights with the force of his will. The most famous Russian psychic is no longer living. Her name was Nina Kulagina. During a long career, she appeared to move all sorts of things through the power of her mind. Now that the media are no longer strictly controlled, there's a vast new audience for the paranormal on Russian television. Every night, a program called The Stars Speak precedes the news with an astrological forecast. The psychic Kasparovsky has even used television to demonstrate his claimed ability to control pain. In this case, during a live operation. There are even commercials that extol the miraculous. During the Cold War, both the Soviets and the Americans carried out research on psychic warfare. At one time, there was fear in the Pentagon about a psychic or psi gap. The Russians claimed some success with psychic mind control, but their evidence was sketchy. So I was interested to find that scientists at the prestigious Institute of the Brain in Moscow had reported measuring psychic effects under test conditions. I arranged to observe the experiment. The data they'd collected in their test seemed promising.
I think we could see the same effect. We see this just before.
It appeared that the psychic had been able to change the brain waves as well as the blood pressure of the test subject. If these results could be confirmed, it would be a real breakthrough for parapsychology. To make sure that the scientists would not know what the psychic was doing, it was agreed that he would be isolated in a remote wing of the complex. Since it was founded in 1926, the Institute has been a national center for the study of the brain, but much of the work done here had until recently been kept secret.
Sit down, please.
The psychic's name was Ignatchenko.
Very good.
According to parapsychologists in Moscow, he was the real thing.
All right, we now have a little over 1 minute before we will start the selection process.
Через чакры, окей?
Accompanying him was Zoya, his assistant. The experiment would last an hour, broken down into 4 15-minute tests. During each test, Ignatchenko would attempt to change either the subject's brain waves or his blood pressure. Or he might be asked to do nothing at all.
Now he reaches inside and he takes one of these at random.
At the beginning of each test, Ignatchenko randomly determined what he would do.
What do we have? That is the null. Okay, so we do nothing for 15 minutes.
For the first segment, Ignatchenko would not attempt any kind of influence.
This will be like a control. So,
The scientists began their measurements, unaware of the psychic selection. For phase 2, Ignatchenko chose a heart symbol. He would try to change the subject's blood pressure.
Повышаем давление.
We are raising the blood pressure,
Поднимает малого таза.
beginning from the pelvis and working along the backbone.
Until there's burning in the backbone. That's enough. There's no need to go any further.
The spreading has already started.
Еще.
Phase 3, the brain. Ignatchenko would try to change brain waves.
Стимуляция.
Stimulation,
Гипофиз.
the pituitary.
Стимуляция гипофиза.
Stimulation of the pituitary.
Окей.
Like that. Good, like that.
The fourth and last test would again focus on changing brain waves.
He will now be very smart. We have stimulated both the right and left brain.
When the experiment was complete, the scientists examined their measurements for changes in the subject's brainwaves and blood pressure. Any variation from normal would indicate a psychic influence.
all right, now let's handle them one at a time. What is our conclusion in test number 1?
If you remember, during the first test, Ignatchenko had done nothing.
Ну, давление вот очень такое.
Может быть давление.
It could be the blood pressure.
Многократные эксперименты по одному не сильно.
The scientists saw a change in the subject's blood pressure.
Then, number 2,
Не сильно.
In test 2, Ignatchenko had tried to change blood pressure.
and now
Может быть двойного влияния.
in
Скорее всего,
test,
второе на мозг.
The effect was on the brain. I think so. Sergei may disagree.
This time, they detected a change in his brainwaves.
Да,
now,
да.
what do we think on 3 here?
Третьем давление,
With some disagreement, they called the next test correctly for the brain.
Maybe we're brain in third case.
Maybe.
Maybe.
Okay. That's brain. What about number 4?
Четвертый.
The last test had been another attempt to change brainwaves.
None. Nothing.
There's none. Okay, there's no.
But the scientists found no change at all.
All right, shall we announce the results then? Let's,
Сказать, что на самом деле было.
let,
The experiment had produced one positive result out of 4. Just what would be expected by chance alone.
The former set of tests.
Not very convincing evidence. So what about their previous claims? I had an idea what might be at work here.
Have some
I suspected that in the past, when they examined their data, they had known what effect they were looking for. In scientific lingo, that the test had not been blinded. They confirmed this, raising the possibility that their previous work had not been objective.
look to see some correlation.
But one of the group believed that the problem lay elsewhere.
I feel strongly that it is impossible to accurately measure these extrasensory phenomena by observing electrophysiological indicators, because the methods are too crude.
But remember that modern methods, no matter how complicated, technical, and technically perfect and advanced, are of no use whatsoever unless it is conducted in a double-blind fashion.
Of course, the results of a single test are in no way conclusive. But one thing I've learned over the years is that scientists, like the rest of us, have an uncanny ability to find what they're looking for, whether it's there or not.
Could see only influence in respect of visual stimulation.
All right. So hopefully you have a little bit more of an idea of how testing is done. There are tests done on everything. There are tests done on pseudoscience, like psychic ability, but then there's also tests done on actual things that we're going to eat, or testing on items that we're going to use on our body. So they can be testing in many different ways. Let's look at correlation. So correlation is observed when there's a statistic variable. So this variable could be the divorce rate of Maine, all right? But then they can have a correlation to something that occurs when there's one variable directly influencing the other. And it can be something very simple that you don't even realize until you start testing. So we have a divorce rate of people in Maine. And then we have the amount of margarine, which is like a type of butter, that has been consumed. And there's a correlation, as you can see, with the divorce rate and margarine. Do you think the margarine or the butter has anything to do with the divorce rate? Probably not. But we can sometimes, when we're testing, find things that are variables that may be very similar to something else in the area. And we can't ignore it, because sometimes they do have to do with what you were observing and looking at as the variable itself. It can't be ignored, but it should be mentioned during the testing if you do find some kind of correlation. So we can have three types of correlations. Positive, which of course means that it's going to increase. Negative means that it's decreasing, or maybe there's no correlation at all. But either way, you still have to mention it in your testing. So we can have pseudoscience, which is false science. And it doesn't matter if you believe in pseudoscience or you don't believe in pseudoscience. That doesn't matter. Right here, we're talking about what science is. And so it has to be mentioned that it is a type and form of science that some people believe in, and that's okay. But it is a type of science, so that's why it's being mentioned today. So these are up here claims that say they are scientific, but they really don't follow the scientific method. So you may make a statement that you were able to walk outside and do something in particular with your hands, and then all of a sudden it starts to rain. You could have that as your pseudoscience, that's your statement, but unless you were able to test it multiple times, have data, have a conclusion, have a peer review, it can't officially be science. And so that's why it's mentioned today. There's just different types of science. So we have the four elements theory, and that is composed of fire, water, earth, and wind. And we even have songs about this. And so pseudoscience was really important in those early ancient days, because this is what got the process of scientific method going. And so they had a theory. It had never been tested. that they had a hypothesis of what they thought was going to happen, and those pseudoscience thoughts then became actual theories that were able to be tested later. And some were true and some were not back then, but at least they were able to do that. So it's important to talk about pseudoscience because it did get things kick-started in a sense in science. All right, fields of biological study. So we have many different branches of science in biology. We have biochemistry. That's like the molecular level of everything. Microbiology, my favorite, studying single-celled organisms, maybe bacteria and virus. Paleontology, studying, the dinosaurs and fossils. Genetics, patterns of heredity and how things are passed on in traits. Taxonomy, how do you classify things by their DNA and their traits? With taxonomy, many insects, this always is very interesting to students, many insects that are currently walking around on the earth or in the ground have never even been identified by anyone yet. So that could be your goal, is you're going to try to identify and name an insect during your lifetime. All right, thank you so much.